Okay, welcome everyone. Good morning. Thank you for tuning in. My name is Kylie Langlois. I am a PhD candidate and research assistant with the New York State Center for Clean Water Technology. And today I am going to be talking to you a little bit about how I've been opening the microbial black box of wastewater treatment. And later on in the presentation, my colleague, Dr. Mian Wang, will take over and discuss some tools she's been using to open the black box. And none of this work would have been possible without the guidance and advisement of Drs. Xinwei Mao and Jackie Collier. Before we get started, I want to note that all the analytical work in this presentation took place at Stony Brook University, which sits on the traditional territory of the Settlecot Nation, and we wish to pay our respects to elders past and present. Here's a brief outline of what we're going to go through today, just to orient you through the presentation. First, I'll give a brief primer on the nitrogen cycle, then put that into context of on-site wastewater treatment, and finally introduce the black box and how myself and Dr. Wang have been opening the black box. Finally, a few points about looking to the future and a take-home message for all of you. So diving right into the nitrogen cycle, this is actually a fairly simplified figure of the nitrogen cycle, and there's still quite a lot going on here. So I'm going to walk you through every component of this. First, I want to introduce you to sort of the main players of the nitrogen cycle. From bottom to top, this is ammonia, nitrite, and nitrate. These are the most abundant biologically available nitrogen species or nitrogen molecules. This means that these react with biological organisms, such as microorganisms or microbes, very readily, and they're very abundant in the environment. Things like harmful algal blooms, like you can see here on the left-hand side, these are actually two pictures taken from bays on the south shore of Long Island. The increase in both the number and magnitude of harmful algal blooms on Long Island have actually been linked to the increased concentration of these biologically available nitrogen species in the water. Now, these harmful algal blooms sometimes are directly toxic to marine life and even small pets. But more often than not, they are harmful because it's such a large amount of organic matter that when they die and sink to the bottom of the water, that the bacteria that start to decompose them actually draw down so much oxygen from the water that they can lead to things like massive fish kills, like pictured here, where the fish actually are not able to breathe in any oxygen from the water and die. So not only is this an environmental problem, this is also an economic problem here on Long Island. It's also a human health issue as well. There's nitrate in the green here can actually interact with hemoglobin in our blood, which is the oxygen carrying molecule in our blood, and makes it very inefficient. So if little babies drink too much nitrate in their water, they can actually develop a blue tinge to their skin called blue baby syndrome, which means they're actually not carrying enough oxygen in their blood. Sort of on the flip side of these biologically available nitrogen species is nitrogen gas. It makes up 78% of our atmosphere and is, for the most part, biologically unavailable, except to a very specialized group of microbes. So there's actually a problem with the nitrogen cycle. In recent decades, there's been increased concentration of nitrogen into the surface and groundwater across the globe for two main reasons. The first is the Haber-Bosch process, which allowed for large-scale nitrogenous fertilizer production. So you have now more nitrogen going into the land to fertilize crops and then being washed off the land from rain or overwatering, and then going into both the surface and the groundwaters. The second reason is increased population density in urban areas, which has increased both human and animal waste, which is a huge contribution of these biologically available nitrogen species to water waste. So one solution actually is wastewater treatment. It's an effective way to remove these biologically available nitrogen species and convert them to the biologically unavailable nitrogen species, nitrogen gas. So ultimately, we want to reduce nitrogen into our waterways. But who's doing all this conversion? That would be the microorganisms, or microbes. The first group I want to introduce you to are the nitrifiers. Now, these are autotrophic, which means they can make their own food, they can make their own carbon. They are aerobic, which means they need oxygen to grow, just like you or me. But they're relatively slow growing and not very abundant. And the important thing here is that there's actually only two groups that can do this. The first group are ammonia oxidizers, which convert ammonia to nitrite. And the second group are nitrite oxidizers, which convert nitrite to nitrate, which is great, but we want to get down to nitrogen gas. So we need another group of microbes, which are the denitrifiers. In comparison to the nitrifiers, they're heterotrophic. They need a carbon source, they need food, much like how we eat food. But they can grow without oxygen, and they're very fast growing, very abundant. And the key thing in here is there's many, many groups that can do this. But ultimately, the denitrifiers convert nitrate down into nitrogen gas, completing our nitrogen cycle. But there's other nitrogen species and processes that I didn't really talk about here, so I want to mention them now. First ones are nitrous oxide and nitric oxide. These are actually very powerful greenhouse gases, and hydroxylamine, which is actually a component of jet fuel and is incredibly explosive at high quantities. So these are actually obligate intermediates created by both nitrifiers and denitrifiers, but they're usually not in the environment for very long or in very much abundance. So for the purpose of this presentation, they're going to largely be ignored. Now, anammox is anaerobic ammonia oxidation. This is different than our typical nitrifier that does aerobic ammonia oxidation. And these are even slower growing than the nitrifiers and even lower abundance, and they're very hard to grow in lab. Nitrogen fixation, this is only done by that very specialized group of microbes that I said before that can actually interact with nitrogen gas and convert it to ammonia. So anammox and nitrogen fixation, since they are very specialized microbes, only really found in abundance in certain environments, again, largely ignored for this presentation. Ammonification you can think of as simple decomposition, and assimilation, ammonia assimilation, you can think of as just normal growth. So now I want to put all of that in the context of on-site wastewater treatment system. Here is a really simple schematic of a current Long Island on-site wastewater treatment system. So anything that goes down the drain, you flush down the toilet, that you wash in your washing machine, will go into a septic tank where solids will settle out, and then your wastewater will then go into either a cesspool or a leaching ring, which is essentially a big concrete ring with holes in the side to allow wastewater to slowly seep out into the surrounding soil matrix. So these systems ideally are for places with a very deep groundwater table and with a very low population density. So the nitrifiers and denitrifiers can actually convert those biologically available nitrogen species in the wastewater to biologically unavailable nitrogen gas. But that's really not the case on Long Island. We have a very shallow groundwater in some places and a very high population density. So this actually can overwhelm and has overwhelmed the capacity of these soil microbes. And in some places, you get a situation like this, where your cesspool is actually in your in the groundwater table, which is the sole source of drinking water on Long Island. A potential solution to this are nitrogen removing biofilters, which is an on-site wastewater treatment technology that the Center for Clean Water Technology is really pioneering at this time. The key thing here is that these are really shallow systems, so they're ideal for Long Island, and they promote microbes within the system, so not relying on soil microbes. Now we know these systems work well. So if you look at these three figures here, this is actually looking at 3 different NRBs over the course of the year and their nitrogen removal. You can see that they remove about 50, 60 to nearly 100% of nitrogen throughout the year. And the dashed vertical lines are simply quarterly sampling that occurred. But what we don't know is why these fluctuations happen. You can see that the 2 NRBs on the ends fluctuate in different ways throughout the year, whereas the middle one doesn't really fluctuate all that much. So we don't actually know what the microbial community looks like in these systems, or actually what they're doing. And this is the black box right here. But if the systems are working well, then why do we even need to open the black box? Well, like I said, we actually don't know what the systems are doing. And we can't design better systems with this very limited knowledge. So I want to zoom in on this NRB from before. So like I said, everything that goes down the drain goes into your septic tank. It is then dispersed onto this sand layer, and by gravity goes into a sand lignocellulose layer. And when I say lignocellulose, I typically mean wood chips, and this can either be very small sawdust-like wood chips or very large wood chips. The assumption here is that certain microbial groups will be concentrated in certain places within this NRB. So this sand layer is thought to provide a lot of space for oxygen to penetrate through the soil, grass, and all the way through the sand layer. So we expect our nitrifiers to be here. Whereas the sand lignocellulose layer may even be contained by a liner, so it's saturated, to have little to no oxygen and a high carbon source from that lignocellulose. So we expect our denitrifiers to be here. So this way, working together, these groups can then transform the biologically available nitrogen species into nitrogen gas. But again, we actually don't know if those microbes are there. So we want to actually test the assumptions of the NRB design. And the way that we did that was we actually went up to the Massachusetts Alternative Septic System Testing Center, or MASTIC for short, and we were able actually to sample a full-scale NRB. So we were able to take a core, so a big metal pipe, we stuck it into the system from top to bottom, and then took the pipe out so that there was a hole left over in the system. And actually, Dr. Xinwei Mao helped to collect these samples by sticking a small tube into that hole, into the system itself. And so we're able to get a sample for about every inch. And right at inch 17, we were able to see some wood chips. So we know we had hit that sand lignocellulose layer. Okay, well now we have all these samples, but how do we open that black box? So the way that I have been doing it is by a metabarcoding technique, where you have a sample, you know there's microbes in there, and then I extracted DNA using a kit and some fancy chemistry. I then sent that DNA out to a sequencing facility where they performed high throughput sequencing, again using very fancy chemistry. And what I got looked like this, just basically DNA sequences, A, C, T, G, and how many times they had appeared in samples. So I can't actually analyze that. So I ran it through various computer programs and used coding as well as worldwide databases to get taxonomic information. And then over the past 2 years, I have been personally curating a database of microbial taxa into their potential functions to get something that looks like this. So for every sequence, I got both the genus and species, so the taxonomic information, and using my database, I then related that to a function. And using the sequencing information, I could tell how many times that sequence was detected in every sample. Now for genus and species, this is analogous to things like cats. So house cats and other small cats are part of the same genus, but they're different species. And so you can see here, this Nitrosomonas on the top row, that could be analogous to the cat species, or the cat genus, and there's very different species. And then conversely, you have things like big cats like lions, which are in a different genus with different species, but both cats and big cats mostly do the same thing. So that's why you'll see here, 2 different genera, which is the plural of genus, are actually both nitrifiers. They have the same function. And so I was able to do that for nitrifiers, denitrifiers, and other functional groups. So diving right into that, looking at the nitrifiers. So to remind you of the assumptions, we expected relatively more nitrifiers in this oxic sand area. And looking at this figure on the x-axis, going left to right is actually going top to bottom if you look in the top right-hand corner of the little NRB schematic. So looking from top to bottom and right at that vertical, black vertical line is right when we hit the sand lignocellulose layer. And the top panel are our ammonia oxidizers and nitrite oxidizers from earlier in the presentation. The left-hand side, the left-hand y-axis shows you the different genus names. They're not really important at this time. And the bubble size is related to how many sequences of that genus was present in that sample. And this is expressed as a %. So in general, this is what we expected. You can see right at that vertical line, which notes the transition into the sand lignocellulose layer, that the nitrifiers simply seem to drop off almost completely. And what's really interesting about this sort of data is that we get everything there is. And so we were able to actually see that there's a lot of redundancy in the nitrifiers. So say for instance, a disturbance took out Nitrosospira, which is the top row of the top panel, and also took out Nitrospira, which is the top row of the bottom panel. There's still other microbial genera that can carry out nitrification. And so we can kind of expect stability even if there's environmental disturbances. Now looking at our denitrifiers. Remember, we expected more of them in the sand lignocellulose layer. And looking at this figure, remember left to right is actually top to bottom, and this vertical black line is the delineation between sand and sand lignocellulose. This is actually not what we expected because they're equally abundant in both layers. And there's some genera, such as Rhodanobacter here, which are actually more abundant in the sand layer. And actually, Rhodanobacter is autotrophic, which you'll remember are like nitrifiers. They can make their own food. So they don't actually need the carbon of the wood chips. And many of these denitrifiers are multifunctional. And what's really interesting again about this data is the large amount of redundancy in this functional group. I want to mention that this is only a fraction of the denitrifiers that were in this data set. Now, I have been leaving out kind of a large step in this process that isn't directly nitrogen related, but is a key step nonetheless. And that is the cellulolytic microbes. So denitrifiers cannot actually interact with the wood chips, the lignocellulose themselves. In fact, they need to interact, they need to have a sort of middleman. And those are bacteria that are capable of degrading cellulose into these volatile fatty acids, which are short and small carbon molecules that denitrifiers can use. So we expect the cellulolytic bacteria to be more abundant in the lignocellulose layer. But in fact, much like the denitrifiers, that wasn't the case. So again, we see from left to right is going top to bottom in the system with that black line delineating sand and sand lignocellulose layer, that they're all pretty much equally abundant in the sand and sand lignocellulose layer. And quite a few of these microbial genera are even preferentially found in the sand layer, where there's no lignocellulose, so we think. But in fact, there's been studies that have shown that lots of cellulose is actually persisting in wastewater up to this point, either by what we eat and then excrete, or even from toilet paper. And so this actually may be supporting unexpected interactions in the NRBs. So to kind of bring this section of the presentation all together, we've seen that we have our nitrifiers in our sand layer. We have our expected cellulolytic bacteria in the sand lignocellulose layer. Then the denitrifiers take those volatile fatty acids and the nitrate from the sand layer and then convert that to nitrogen gas, which again is our desired end product. But we have this other group of cellulolytic bacteria, which may be interacting with toilet paper or some other form of cellulose in this aerobic sand area, which then in turn may be supporting other microbes involved in iron and sulfur cycling, which in turn, those are the microbes that may be supporting these autotrophic denitrifiers. So it's not so black and white as we previously thought. Now, I've been talking a big talk about this metabarcoding technique, but there are some limitations. Mostly that this can really only determine potential function. I only know the taxonomy and what the literature has told me that these microbial genera can do. And this is especially tricky for microbes that are multifunctional or they may be inactive. So they may not actually be doing the function in this sample that I detected them in. But we can count these functional genes, which are actually doing these functions with quantitative PCR that Dr. Mian Wang will be discussing. And this gives us a more accurate picture of different aspects of the black box.
Hello, everyone. My name is Mian Wang. I'm a postdoc working with Dr. Xinwei Mao on the novel treatment technologies for decentralized wastewater. Next, I'll talk about the quantitative tools we are using for microbial community analysis in the NRB systems. Quantitative real-time PCR, also called real-time PCR or qPCR, is a PCR-based technique that can amplify and also quantify the target DNA in real time. qPCR has been widely used in environmental microbial studies as a tool for detecting and quantifying microorganisms of interest. It can help us to have better understandings of the complexity of wastewater microbial communities. Despite the important role that microorganisms play in nitrogen removal, few studies have investigated the nitrifying and denitrifying microbial communities in on-site wastewater treatment systems by qPCR. For our NRB projects, the first step is to collect sand and wood chip samples from the columns. Then we will extract DNA or RNA from the samples for the downstream molecular assays. By using primers, qPCR can provide the absolute quantification of the target genes, thereby selectively estimating the population of target microorganisms. However, there is a wide variety of microbial groups in real wastewater, posing a great challenge for the primer selection and optimization. We adopted the molecular method for this key process to establish selectively primers for our target microorganisms. Finally, we'll select the primers with the best amplification efficiency and coverage to develop the qPCR biomarker platforms for quantification. Applications of qPCR quantification would help us gain more in-depth insight into microbial communities in NRB systems under both oxic and anoxic conditions. Such quantitative information facilitates linking the nitrogen transformation levels with the abundance and activities of the functional microbial species. Besides that, mathematical models could also be optimized with the data from qPCR to develop systems-level predictions of the nitrogen transformations in an NRB. The application of qPCR could broaden our understanding of NRB systems at the molecular level, and also advance the prospects for better process control and design in the future. This page shows the partial nitrogen pathways involved in our NRB systems. Kylie already talked about this in general, so I will just focus on the enzymes and the functional genes in these processes. The first is the nitrification process. Nitrification is a two-step biological process comprising nitritation and nitratation, which are performed by ammonia oxidizing bacteria, AOB, and the nitrite oxidizing bacteria, NOB, respectively. The commonly used biomarker to investigate this process is the AMOA gene. And in denitrification process, nitrite to nitric oxide reduction is catalyzed by two functionally equivalent types of nitrite reductases. One is the copper-containing enzyme, encoded by NRK. The other is a cytochrome D enzyme, encoded by NYS. These two genes are commonly used as nitrite reduction markers to study the denitrifying bacterial community. Let's now turn to the two case studies. The first project is collaborated with Zara on the lab scale NRBs. In this study, the effect of environmental and operational conditions on the nitrification process are under investigation. I analyzed the microbial community and functional gene distribution at different depths using molecular tools. The second project is collaborated with Sway on the next generation NRB with pre-aeration and internal recirculation to investigate the nitrogen transformation. I estimated the abundance and the distribution of functional genes on both nitrification and denitrification processes. Therefore, the overall goal of this series of studies is to investigate the nitrogen transformations in NRBs with different configurations by quantifying the abundance of functional genes using qPCR. The first study was conducted to help in designing an optimized configuration and operation strategy, which can shrink the system size and make NRB more suitable for areas with shallow groundwater tables, like Long Island. The current NRB sand layer has a depth of 45 centimeters to ensure the sufficient hydraulic retention time for complete nitrification. Therefore, we use 3 lab scale sand columns with various depths of 15 centimeters, 30 centimeters, and 45 centimeters to investigate the effect of operational changes on the nitrification performances. These 3 columns were filled with the aged sand matrix, which was taken from an in-ground NRB that had been continuously receiving septic tank effluent at the Massachusetts Alternative Septic Systems Test Center. We increased the nitrogen loading and hydraulic loading at stage 3 and 4 respectively, and at stage 5, real wastewater was fed to the columns to investigate the system performance. Thus, the application of synthetic or real wastewater was used to demonstrate the ability of sand columns to achieve high levels of nitrogen removal. We also placed the oxygen sensors at 4 depths in the columns to monitor the DO levels, which was kept below 2 microgram per liter by purging with nitrogen gas. In this project, we have detected 16 sand samples in total. Homogeneous samples were the aged sand matrix, and they were taken at the beginning of the experiments. At the end of stages 3 and 4, microbial samples were taken from the bottom of each column to compare the microbial community response to the operational conditions at each depth. The column were dissected at the end at all experiments in order to illustrate the vertical profile of microbial community. The right figure shows the dissection of the columns. Samples with orange colors were quantified by the qPCR assays. These 4 figures show the quantities of bacterial 16S rRNA, AMOA, NRK, and NRS genes at different stages and depths. The x-axis lists the depths of the sand columns, and the y-axis represents the log gene copy numbers per gram. The total bacterial biomass was measured as the quantity of 16S rRNA genes. We can find that at all stages, the top layers of columns have the highest gene copy numbers. The abundance of 16S rRNA, AMOA, and NRK genes declined with the depths, specifically at stages 4 and 5. The downflow pattern enriched the microorganisms near the surface, which was consistent with previous observations in sand filters. The AMOA gene is often regarded as the marker of ammonia oxidation, in which ammonia is oxidized to nitrite. As shown in the upper right figure, the gene copy numbers of AMOA in these 3 columns were higher than that in the startup homogeneous sand. The results indicated that our sand columns and the aerobic environments were more favorable for AOB to grow and function. Combined with chemical analysis, effective nitrification could be achieved at 15 centimeters of the columns at all stages. Effective nitrification at shallow depths is important in the design of in-ground on-site wastewater treatment systems, especially at regions with shallow groundwater tables. The footprint and the cost of NRBs could be reduced by decreasing the depths. The bottom 2 figures show the absolute abundances of NRK and NRS, which are the 2 functional genes involved in the denitrification process. The quantities of NRS gene were below the quantification limit in almost all sand samples. These results were consistent with previous studies in wetlands, which showed that denitrifier communities was dominated by NRK-containing denitrifiers over NRS-containing denitrifiers. Although the 2 nitrite reductase genes are functionally equivalent, bacteria with either gene have a preference for certain environments. Denitrifiers with the NRS gene prefer stable environments with constant and anaerobic conditions, while denitrifiers with the NRK gene are less sensitive to fluctuating oxygen concentrations. The 16S rRNA quantity at stage 5 was considerably higher than that in homogeneous Finland sand, which was not observed at stage 3 and 4. It can prove that real wastewater brought new microbial community to the systems. In addition, The microbial community originally present in the aged sand matrix could sustain efficient nitrification when synthetic wastewater was applied. If the NRBs receive diluted influent with low biomass, they would still be able to maintain their performance. Besides the NRB 1.0, we are also exploring the possibility to enhance nutrients removal efficiency and decreasing the footprint of current NRBs by designing novel biofilters. As shown in this figure, NRB 2.0 system consists of 4 major parts: the pre-aeration system, nitrification column, intermediate denitrification column, and the final polishing denitrification column. All nitrification columns are filled with C33 sand, and all denitrification columns are filled with wood chips. The pre-aeration system contains 1 aeration tank with an air pump. Septic tank effluent is mixed with the recycled effluent from the intermediate denitrification tank at 1:2 volume ratio, and is continuously pumped to the nitrification column for further treatment. The air pump is an optional design, which is easy to turn on or off to control the DO concentration. The nitrified effluent from the nitrification column is then distributed to the denitrification columns. In there, nitrate is denitrified to nitrogen gas with the carbon released from the wood chips. The effluent from the final denitrification column is discharged to wet wells, where effluent samples will be collected for water quality analysis. For the next generation NRB project, we have collected solid samples in nitrification and denitrification columns. You can see from the figures, 5 sand cores were taken from each nitrification column, and 1 wood chip core was taken from the center of each denitrification column. Each sand core from nitrification columns was sectioned into 4 samples, corresponding to 4 different layers. And each wood chip core was sectioned into 3 samples, corresponding to 3 different layers. The sand samples at the same depth interval were mixed into 1 samples for further qPCR assays. Now let's move on to the microbial results of the NRB 2.0. The x-axis shows the different layers of the columns. The black, red, and the blue bars represent the log gene copy numbers of functional genes in nitrification, intermediate denitrification, and the final denitrification columns, respectively. As you can see, the qPCR results of NRB 2.0 also confirmed our conclusion of the lab-scale NRBs. The abundance of all target genes decreased with the depth in the nitrification sand columns. That's because the nitrification columns in both studies are operated at downflow patterns. But for the intermediate and the final denitrification columns in NRB 2.0, they are operated at saturated conditions and also upflow patterns. So the abundance of target genes not changed with the depth. The functional microbial species were evenly distributed in the denitrification columns. We can also find that the concentrations of NYS and the NRK genes were similar in the nitrification columns, and they both contribute to the denitrification process. The next steps of this series of NRB studies would be to continue to investigate the abundance and the distributions of 16S rRNA and the functional genes in NRBs with different configurations. The quantitative information will aid in evaluating the performance of different NRB systems. The size of the microbial population that carry out the nitrification and denitrification, their presence effluent and other wastewater properties may help us to explain differences in performance. Based on the solid foundation for nitrification and denitrification studies in NRBs, it will be easier to expand the qPCR biomarker platforms, include the RNA, to investigate nitrogen transformation of different pathways, like nitrous oxide formation. The nitrous oxide is a greenhouse gas and also an intermediate nitrogen product produced in the denitrification pathway. We'll combine the qPCR data with gas analysis to assess whether these biofilters completely transform nitrate to nitrogen gas. Finally, we'll also integrate the microbial data with mathematical modeling for system performance evaluation. With the help of molecular tools, we can definitely open the microbial black box in the NRB systems. So now I'll pass you over to Kylie for the conclusion of these presentations. Thank you.
Thank you, Mian. And just to finish up the presentation, kind of so what? You know, you watch this whole presentation and why? Well, actually knowing the relative abundance of these different microbes and the genes through qPCR that Mian has done, not only have we validated NRB design, but we've also shown that, you know, we can have some confidence in the nitrogen removal of these systems. And this also lets us inform decisions for future NRB design, the next generation of NRBs, to improve nitrogen efficiency and decrease the cost to the homeowner, so we can actually get these systems out to as many homeowners on Long Island as possible and reduce those biologically available nitrogen species into our waterways, which is the ultimate goal of this system. And with that, I just want to thank everyone here for coming. I would like to thank the New York Center for Clean Water Technology for supporting myself financially and through a lot of research advice, and also for hosting these seminar series. There will be a few more of these throughout the summer. You can click on the website listed here. If you would like to follow the Center for Clean Water Technology, Dr. Collier, or myself on Twitter, our Twitter handles are there. And finally, I would like to thank the School of Marine Atmospheric Sciences, which myself and Dr. Collier are a part of, as well as the College of Engineering and Applied Sciences that Dr. Mao and Dr. Wang are a part of. And finally, Stride, which has supported me financially for a number of years as I did this work. And with that, I'll take any questions from the chat or by raising your hand. And again, thank you so much for listening. Thank you. Okay. Just looking in the chat right now. Let's see. So Ed asked, does the sand sawdust layer have a greater tendency to plug? And that actually, yes, has been an issue in the past, but only for one of the systems. That was a saturated system that was lined with an impermeable layer. And they think what happened is too much rainfall. So that is definitely something that we're looking into, of different types of wood chips to use, different sizes of wood chips to use to reduce that plugging. Ed also asked, in sequencing data, did we find PAOs? I'm assuming those are phosphate accumulating organisms. Yes, we did find a few of those, not in very much abundance. Other people in the center have worked on phosphorus work in the NRBs. And if I can remember, Dr. Laura Wehrman and her students have looked at this and shown that NRBs actually do remove nitrogen. They do a pretty good job of removing phosphorus. And so we definitely have seen those microbes in there. Ed, again, had a lot of questions. How How long does the cellulose perform before nitrate removal tails off for Long Island septic systems? So the literature has shown that wood chips can actually support the cellulolytic bacteria, which then support the denitrifiers for upwards of 20, 30 years in both the field and the lab. And so far on Long Island, I don't want to put my foot in my mouth and give the wrong numbers, but I know that for the pilot systems we have going on Long Island currently, there hasn't been any sort of tailing off due to the wood chips. And Hillary posted where the recordings will be. Thank you. And does the septic tank effluent require an effluent filter? So there has been some talk of polishing systems, which may be what you're referring to, Ed. And so far the goal is to not really need that. Is to basically have the nitrogen coming out of these systems to be below 10 milligrams of nitrogen per liter, which is 50%, which is 50% of what Suffolk County requires, which is 20 milligrams of nitrogen per liter. So we're trying to do even better than the Suffolk County limits. And so we're hoping that then, we can hit that goal. And for some systems, we have hit that goal really consistently. And then we wouldn't need a filter. Oh, Christopher, thank you for clarifying. Effluent filter of the septic tank. So the septic tank effluent, excuse me. Yeah, so I'm not sure on that one. That's more on the engineering side. I think there is some sort of filter. Typically, I know the Mian mentioned work that is actually ongoing in a research trailer that we have that is receiving real wastewater. Oh, Stuart says no filters from the septic tank. So he knows more than I do on the design features. And someone asked the cost of the lowest price of the system. And again, I don't know that specifically. Oh, sorry, there's, okay, I have gotten private information that there is a filter, an effluent filter before going into the tank. So Chris, you were correct. And so these are all things that I'm not involved in. I mainly look at after the fact to see if the design is working. And on the cost, again, that's something you'd have to contact the center for. Let me flash back to the website and the email. I know that we have been working very closely with Suffolk County to make these systems as affordable as possible.